Hello everyone, my name is Narendra Ashikin, number metric 102901, and today I would like to present my poster under title Degradation of Polyethylene Terephthalate or PET plastic by wastewater bacteria engineered via conjugation. First, let me begin with the introduction. Plastic pollution has become one of the biggest environmental problems worldwide. Since 1950, more than 4,900 megatons of plastic waste have been dumped into the environment, and this amount is expected to triple by the year 2060. Among this plastic, PET is commonly used in bottles, food containers, and coffee cup lids. Unfortunately, PET is very resistant to natural degradation and can break down into harmful microplastic that contaminate water system and food chains. Scientists have discovered enzymes such as phosphatase that are capable of degrading PET efficiently under different environmental conditions. However, introducing foreign laboratory bacteria directly into wastewater environments is often unsuccessful because native microbes usually outcompete and eliminate them. Therefore, this project used a different strategy. Instead of adding foreign bacteria, native wastewater bacteria are genetically engineered using conjugative plasmid. In simple terms, the plasmid acts like a delivery system that teach environmental bacteria how to produce the phosphatase enzyme and degrade plastic naturally. Now, moving on to the objective of this study, the first objective is to engineer native wastewater bacteria by transferring a conjugative plasmid that carries the phosphatase gene. The second objective is to evaluate the efficiency of the purified phosphatase enzyme in degrading commercial PET film. And the third objective is to measure the degradation of post-consumer PET materials, specifically coffee cup lids, using conditioned media produced by these engineered bacteria. Next, I will explain the methodology used in this project. The first methodology is plasmid and donor strand construction. A plasmid containing the phosphatase gene was constructed together with an M-cherry fluorescent marker. The M-cherry marker produces a pink color, allowing successful bacterial transformation to be easily identified. Step 2 is engineering wastewater bacteria. Raw wastewater samples were filtered to obtain native microorganisms. 24-hour filter mating process was then performed, where donor bacteria transfer the plasmid into the wastewater bacteria through conjugation. After selective isolation, successful engineered strands appear as pink colonies because of the M-cherry marker expression. Step 3 is enzyme purification. The engineered bacteria were cultured and arabinose was used to induce the production of phosphatase enzyme. The enzyme was then purified using nickel anti-A affinity chromatography to obtain purified phosphatase for degradation experiment. Step 4 involved degradation assays. Two degradation systems were tested. Track A used commercial PET film treated with purified phosphatase enzyme at 50 degrees Celsius for 4 days. Track B used conditioned media from engineered wastewater bacteria to degrade post-consumer PET coffee cup lids at 50 degrees Celsius for 5 to 7 days. Finally, step 5 is surface and structural analysis. The degradation process was evaluated through mass loss measurement using an analytical balance, surface observation using SEM, and structural analysis using UV-Vis spectroscopy and DSC test. Now, let us look at the expected outcomes. The results are expected to show that purified and raw phosphatase produced by engineered wastewater bacteria can successfully degrade commercial PET film by around 40% within 4 days. For post-consumer coffee cup lids, approximately 1.3% degradation is expected after 7 days, together with visible surface damage such as pitting and cracks. In the figure shown here, the untreated lid remains smooth, while the arabinose treated lid show obvious degradation after incubation at 50 degrees Celsius. Most importantly, this project demonstrates a proof of concept of genetic bioaugmentation. This means environmental bacteria can be genetically reprogrammed through conjugation to actively produce enzyme and degrade microplastic directly at the pollution source. In conclusion, this study combines biotechnology, environmental microbiology, and genetic engineering to provide a sustainable and innovative approach for PET plastic degradation using native wastewater bacteria. Thank you. That's all from me.